1. Today I will talk about how the study of gene expression in living organisms is conducted. In particular, I will talk about the transcriptome, or the comprehensive analysis of gene expression using a technique called bioinformatics. I think researchers may say many different things about what bioinformatics is. My understanding, as I wrote here, is that bioinformatics is the taking huge amounts of data, often genomes, transcriptomes, metabolomes, etc., produced by biological research from species, individuals, and populations. It is then the use of computer analysis techniques to analyze the data in order to gain new knowledge from the seemingly miscellaneous and massive data. The actual data handled are mainly numerical data such as measurements, textual information such as nucleotide sequences, and microscope images and movies. Since we will be discussing the transcriptome, numerical data representing gene expression levels will be the main target. As for the fields in which it is used, it is actively used not only in basic research at universities and research institutes, but also in the medical field, in companies developing pharmaceuticals, and in the agricultural field, such as in breeding. Slide 2. Transcriptomics is an analytical method that aims to comprehensively understand the behavior of the entire RNA working in cells, tissues, and individuals, as well as the expression levels of individual genes. The flow of information in an organism consists of the genome, the transcriptome, which is the RNAs transcribed from the genome, the proteome, which is the translation products from the RNAs, and the metabolites produced by the enzymes in the proteome. The transcriptome is analyzed using DNA chips or, more recently, next-generation sequencing technology, although the term next-generation may not be appropriate since it has been used for more than a 10-year period. When using next-generation sequencing, RNAs extracted from tissue samples are reverse transcribed to create complementary DNAs, which are then fragmented, sequenced on a next-generation sequencer, and analyzed using computer and statistical methods to determine levels of gene expression. The expression data can then be used to examine how gene expression differs from sample to sample. The expression data can also be used to identify the biological characteristics of the sample. Slide 3. The most widely used next-generation sequencers today are instruments manufactured by Illumina. Each sequence data output from the sequencer is called a read, and although one read is as short as 50 to 200 bases, the number of reads can be as large as several million or more. The format of this output digital data is called FASTQ. The format is like this blue section. The first line starts with @ the third line starts with +, both followed by the name of the read, the second line is the sequence of the bases, and the fourth line is the symbol for the certainty, called quality, of each base. 4 lines are 1 read, so counting the number of lines in this file and dividing by 4 will give the number of reads. The data size is so large that it is virtually impossible to process on a PC with a word processor and mouse. Slide 4. The output is a large number of reads derived from RNAs. Therefore, each and every one of those reads is a sequence derived from the RNA of a specific gene encoded somewhere in the genome. Therefore, by comparing the nucleotide sequence of each read with that of the genome, it is possible to determine which read is derived from the RNA of which gene. For example, this figure shows a part of the genome. If we paste the obtained reads into the corresponding regions, we can see that many reads are attached to this and this parts of the genome. This indicates that there are genes actively transcribed here and here, and by counting the number of reads, we can also determine how actively transcribed the gene is. By comparing how many reads are attached to this gene in two samples, we can see how differently expressed the gene is in the two samples. Such an analysis can be done simultaneously for all the genes that an organism's genome encodes, which can be tens of thousands of genes. For non-populous organisms, such genomic information is not available. However, since there are many reads obtained from the RNA of the same gene, we can rely on the overlap of these sequences with each other and link them together to reconstruct the original full-length RNA sequence. This process is called de novo assembly. Using the de novo assembly sequence obtained in this way instead of the genome, it is possible to determine how much of each RNA is made, i.e., the level of transcription. Furthermore, by comparing the sequences of the constructed RNAs with the database, we can determine what kind of proteins they encode and what kind of biological phenomena they are involved in. Slide 5. Specific softwares are used for computer analysis of the data. The top left is software used with the operating system called Linux, which handles huge files, and you enter commands for the work with the keyboard in the black area. Sometimes a program called a shell script is written to automate the process. This may be difficult for those who only use Windows PCs. You may also use programming languages such as Python or R to automate data processing. Bioinformatics researchers around the world have created excellent analysis software and released it free of charge. Some of such software is available in a web interface as well as in Linux. Here are some sites called Galaxy, IDP, and David. Slide 6. Let me explain how to perform a transcriptome analysis by following the actual procedure. First, use the Galaxy website. This is the original site, and there are similar sites in the EU and Japan. The available analysis tools are slightly different from each other, so you may want to use different sites depending on what you are analyzing. Once logged in, a list of available tools is displayed in the left column of the main page. Here you can search for tools by keyword. Clicking here will bring up a sub-window where you can use drag and drop to upload a data file, such as a FASTQ, to the server. When the job is complete, it will be displayed in the right column. Slide 7. Let's try de novo assembly. When performing transcriptome analysis on non-model organisms, although FASTQ files are obtained, there are no reference sequences to compare them to, so de novo assembly must be performed to create your own reference sequences. Here, we call the tool Trinity, which I think is most commonly used for de novo assembly of eukaryotic RNA data. Here you specify whether the FASTQ file you uploaded is single-ended or paired-end. In most cases, it is paired-end. Then select each FASTQ file. Finally, press the button here to run the analysis. This may take overnight or longer. When it finishes, you will see it in the right column. Download it, review its contents, and if necessary, do some pre-processing on your own PC for the next analysis. For example, removing short sequences. Slide 8. There is also a tool to determine what amino acid sequence is encoded in each of the tens of thousands of de novo assembly sequences obtained. By searching for the keyword transdecoder, you can find this tool, which will analyze a file of de novo assembly sequences and output the nucleotide sequence and the estimated amino acid sequence of the predicted translated region for each sequence. After downloading these sequences and comparing them with the protein database using the tool BLAST, it is possible to estimate what kind of protein each sequence encodes and what function it has. Slide 9. Each of the de novo assembly sequences created above corresponds to a predicted mRNA sequence, or gene. These are used as reference sequences to which reads from the FASTQ file are attached one by one. The number of reads attached to each reference sequence is counted. The tools Bowtie2 and HISA2 are available for this purpose. You call the tool with a keyword search, specify a pair of reference sequence files and FASTQ files, and run the calculation. This is also a long calculation, taking several hours per pair of FASTQ files. In addition, it is usually necessary to repeat the experiment about 3 times per sample and analyze multiple samples. When the calculation is completed, the tabulated result files can be downloaded from the right column. Slide 10. The next step is to install and run the software on your own PC, not on Galaxy. I like this software called Tablet, which visualizes the results of Bowtie2 and HISA2. This software allows us to visually see where each read is attached to each reference sequence. Slide 11. And you can also see the total number of reads attached to each reference sequence. Copy and paste this table into a spreadsheet such as Excel. Do this for each pair of FASTQs and join the table together horizontally to obtain a matrix table of expression levels for each gene in each sample. The vertical direction is the genes and the horizontal direction is the repeats and samples. The actual merging is done using the merge function in R. To standardize the data, the number of reads can be converted to the number of reads on each gene per million total reads and per 1,000 bases of gene length. This value is called RPKM, meaning 1 million reads per kilobase. Slide 12. If your research subject is a model organism, you do not need to perform de novo assembly yourself. There is a reference sequence, i.e., the genome sequence, and there is an annotation file that shows which exons of which genes are in which regions of the genome. With such files, it is easy to add reads from the FASTQ files to each gene using HISA2, format them using SAM tools, and count the number of reads for each gene using stringtie. The entire process up to this point can be done on Galaxy. For example, this site is called Plant Ensemble and contains data for a variety of plant species, even plant species that are not necessarily model plants. The DNA FASTA file is the whole genome sequence file and the GTF or GFF3 file is the annotation file. Slide 13. Following the above procedure, multiple samples are sequentially analyzed and the read counts are joined to produce a table like the one shown here. Vertically are all the genes of this organism, tens of thousands of genes in the case of eukaryotes. And horizontally are the samples and repeat results. In the example shown here, the control group n equals 4, i.e., the experiment with 4 repetitions, and to the right are the results of the treatment group with 4 repetitions. Once you have such a table, you can then use statistical methods to reveal how the two samples differ, and which genes have increased or decreased in expression level by how many fold, and whether it is statistically significant or not. Slide 14. Here, let's upload the table file to the IDP website and analyze it. Slide 15. Shown here is a tool called Edger, a tool designed for R. It will normalize and log transform the uploaded table so that you can compare across samples. You can download the resulting file here. Here is an example of the result. Before the pre-processing, the total number of reads was uneven among samples or repetitions. After the pre-treatment, this is a box plot of the expression levels of all the genes, which are now well balanced and thus can be compared to each other. Slide 16. You can easily make a heat map. You probably see heat maps often in papers on biology. The vertical direction is for genes, and the horizontal direction is for samples and repetitions. Gene expression is colored red when gene expression is high and green when gene expression is low. Above is a cluster analysis of similar gene expression, with the 4 left columns being the treatment group and the 4 right columns being the control group. Since this is a repetition experiment, it is not surprising that the 4 are grouped into a single cluster, but we can visually confirm that this is the case. Vertical cluster analysis has also been performed in genes showing similar expression patterns are located close together. Slide 17. This is a principal component analysis, or PCA, conducted based on the expression levels. The control and treatment groups are clustered separately. On the right is a relatively new clustering technique used for a similar purpose, called TSNE. This one has more compact clustering than the PCA results. This type of analysis can be easily performed. Slide 18. Genes whose expression levels changed between the control and treatment groups are extracted. DESeq2, a tool designed for R, is available. Here, you can specify the adjusted p-value threshold for multiple testing. Here, you can specify how many times higher or lower the expression level of the gene is to be extracted. A bar graph will then be generated showing how many genes have increased expression levels and how many have decreased expression levels. From here, you can download the values of the fold change and adjusted p-value for all the genes, so that you can later set the threshold as you like and extract the differentially expressed genes. Slide 19. This is another clustering technique, a type of network analysis called WGCNA. This is also a tool designed for R. If you visualize the results using another visualization program named Cytoscape, each ball represents a gene, and genes with similar expression patterns are located in close proximity to each other and connected by lines. It is possible to find the genes that dominate this network, such as transcription regulators. Slide 20. Once a group of differentially expressed genes has been extracted, investigate what biological characteristics they have. Biological characteristics include, for example, what reactions they catalyze, what metabolic pathways, such as photosynthesis, they are involved in, and what biological phenomena they are involved in, such as high-temperature stress resistance. This type of analysis is called gene ontology enrichment analysis. Shown here is a portion of a textbook I wrote for another lecture, the subject of Fisher's exact test. I will speak as written. Gene ontology, GO, is a concept which attempts to classify many genes in many organisms by a unified term, GO term, based on their functions and subcellular localization. In the case of model organisms, a database is available to identify which GO term is assigned to which gene. When cells of an organism were treated with compound and the expression levels of all genes were measured by RNA-seq, RNA-seq is the transcriptome analysis I'm talking about. When the RNA-seq was conducted, 507 genes showed a significant increase in expression levels, 5 of which were assigned the GO term cadmium ion response. On the other hand, the genome of this organism encodes 25,119 genes, of which 62 have been assigned the same GO term. This is expressed in a 2x2 contingency table as follows. Fisher's exact test allows you to determine whether there is a significant increase in the number of genes assigned this GO term to this gene group, compared to the reference gene group. In the case of this data set, it is also inferable that compound A, which was used in the treatment, is likely to have properties related to cadmium. Slide 21. There is a site that automatically performs such enrichment analysis for all GO terms. This site is called David. Paste the list of genes whose expression levels are significantly elevated into the site and specify the species to be studied. Slide 22. For Fisher's exact test, a reference data set, called a background, is required. Given a species, a group of all the genes encoded by its genome is used as a background. Then execute the program. Slide 23. Within a moment, you will see the results of the enrichment analysis for all of the GO terms. Let's take a look at a magnified part of it below. This is the result of the GO term cell adhesion. In the list of genes given to the program, there were 42 genes that were assigned this GO term. Click here to see the names of those genes. This is the statistical significance, or p-value, and next to it is the adjusted p-value for multiple testing. Slide 24. If the genome data is not prepared for use as a background, the analysis cannot be performed for that organism. David supports only a small number of species. In addition to David, many other sites provide a function for enrichment analysis. For example, this AgriGeo version 2 is a great site for botanical and agricultural researchers, as it is specialized for many plant species in the field of agriculture. If your organism is unfortunately not supported, you can use your own de novo assembly as a background to run enrichment analysis on your PC. It would be difficult for beginners, though. Slide 25. Let me summarize the story so far. You can do a lot of analysis on web like this. These tools that run on the web may actually be programs that run on Linux or on R. In many cases, you actually have to use R or other software to transfer data between programs. In many cases, you need to read the manual of each tool to understand what it is doing and whether you are using it properly. To understand this, knowledge of data analysis such as Linux, R, statistics, etc., is also necessary. Conclusion: the bioinformatic gene expression analysis is an analysis method to find new knowledge from big data which looks too complicated at first glance. Web tools are useful, but that does not mean that Linux, R, and statistics are unnecessary, so you should learn how to use them. That is all. Thank you very much for your attention.